That's really good. Okay, let's get started. And thank you so much for coming over. This is the first seminar, Hanover seminar series this semester, and we changed the format quite a bit. So I was kind of nervous to how many people we have. So I put my best effort, like walking around, put the poster just everywhere. So I'm pretty happy that we got a lot of people. And it's my honor to introduce Dr. Eagar. And Dr. Eagar joined MSU 1.5 years ago, along with the Dr. Christopher Chriswood, hosted in the Department of Plant Biology. And I have been hosting a forest ecology journal discussion group. And I can remember that the first time he showed up in 1.5 years ago. And three things. One, is it the echoing?
Somebody was echoing.
Yeah. Can you turn it off?
It's okay.
I won't say who it is, but it's not you.
Okay, that's good. So, three things that I noticed. Okay. The first, he talks a lot. And second, he's passionate. And third, he knows a lot. So it's really, really impressive that his knowledge covers all kinds of different disciplinary of the ecology as well as depth of the knowledge. And I'm gonna give you heads up. He's gonna talk a lot. He's passionate and he knows a lot. And it's all yours.
Well, thank you, Aki. Thanks for the invitation. I'm really excited to talk to you all today, especially kicking off the first Hanover seminar speaker here for this semester. It's a lot of pressure, so I hope to live up to it. And like Aki said, we're going to cover a lot of ground in this talk, and I do like to talk a lot. We'll save questions till the end, and I'm also going to be eating lunch with many of you, but I don't have much to do for the rest of the day. So if you find something particularly interesting, either about the whole presentation or a particular area of the talk, let me know and we can just chat later at some point today. And yes, I have a lot of words on the screen, but we'll go ahead and get started here. I do want to take time to thank everybody I've worked with over the years. What I'm going to be showing you guys from both the background theoretical perspective and the data that I've worked on is really the culmination of about 10 years of work that I've been doing predominantly with Chris Blackwood, who is my PhD advisor at Kent State, but also Kurt Smemo and Rich Phillips, who are at Indiana University in Skidmore, of which Rich is coming to talk here in about a month about how his work relates to this and the biogeochemical side of what we're going to be exploring today. And then Andrea Case, who is on my committee, who helped form a lot of the theoretical future directions that we're moving to with this work. I also have a pretty great group of lab members that I work with. Princess, Megan, and Sarah have all contributed in various ways to these projects over time. And then again, we've been doing this for about 10 years for a lot of this work. And we've had, I don't know, I can't even count the number of undergraduate students we've had work on this, from the lab work to the field work and to lab meetings there. I've also been lucky to receive funding from a bunch of different sources to help fund a lot of this work. So thanks to the NSF and the DOE, in addition to some internal scholarships at Kent. Okay, so to get us started, I want everybody to picture their ideal forest. I'm sure it looks something like this, right? We have trees. Trees are necessary components of a forest. But there's also understory plants, and there's a fairly high diversity of leaf litter that is spread out on the ground. There's maybe some twigs and some deadwoods and things like that. But overall, it's a very pleasant place to go hike, and it's also a very pleasant place to have to go sample and work. And it's one of the reasons why I enjoy working in these systems. But I see things a little differently than many of you probably do, because this is really what keeps me up at night. What's going on below the surface of the soil, what's down there where the roots are at, what we describe as the hidden half that you probably wouldn't have any reason to pay attention to unless you're looking down the whole time you're hiking. So we know here that there's a lot of roots that interact in the soil environment. Soil itself is this really chemically complex and diverse environment that is full of microbes and different nutrients in different forms. There's a lot of water, there's competition going on. So how all of these things integrate from populations and communities to ecosystem scales is really kind of what drives the work that I do there. So I try to study this so that way we can explain this and try to figure out maybe which trees are growing where and what the long-term consequences are for these plant-soil interactions in forests as they relate to forest communities. So we are going to try to answer and tackle three broad questions with my talk here today. And I've color-coded them to correspond to the different sections of the talk. There's a lot of background and theory that I want to share with you all. A lot of it is hypotheses that I'll then show some evidence for in the middle portion of the talk, and then we'll wrap up by discussing some future directions. But the first question we're going to ask is, how can different tree mycorrhizal associations generate different plant soil feedback outcomes? And I'll discuss what each of those components are here when we get into the background. Next, assuming I can convince you all that different tree mycorrhizal associations are important for this thing we call plant soil feedback, we're going to ask the question of, can these interactions actually affect tree and soil microbial community dynamics through this idea of spillover effects? And we'll talk a lot about that in pretty close detail as well. Finally, to go under some future directions, if we have these interactions that are affecting plant soil feedback and these communities truly are dynamic, what are the consequences of our understandings for this in a global change context there? So to start our background here on this question, I need to briefly talk about what mycorrhizal associations are. I'm sure some of you are probably pretty familiar with them. Others might not necessarily know exactly what they are or how there are different types, so we'll cover that. I'll discuss plant soil feedback pretty specifically and technically because it's really cool and it's also really important. And then I'm briefly going to touch on this idea of conspecific density dependence. And this is a way that we can look at populations and communities of plants and specifically trees and determine if plant soil feedback, which is a process, is occurring and to what degree is it occurring. So this is kind of the physical manifestation of these mechanisms and processes here. I'm going to go ahead and preemptively get my laser pointer on here. Just when I go reaching for it later, I'll know where it's at. Okay, so mycorrhizas or mycorrhizal associations are symbiotic relationships between plant roots and fungi in which plants exchange photosynthetically derived carbon, sugars, to these fungi that live in their root systems, who are scavenging around the soil environment, taking up mineral nutrients and water, and exchanging it for that carbon they're getting from their host trees. So they're fairly ubiquitous and they're very critical and they're very, very old from an evolutionary standpoint. I think they're personally really neat because if you look at this pine seedling we have here, you can notice its root system is not all that extensive. But with this mycelium from the mycorrhizal fungi that are growing off of it, it is able to explore a much wider array of the soil environment than it would otherwise without this fungal symbiosis. So globally, about 92% of all plants form some type of mycorrhizal association. They're incredibly widespread, and there are a few different types. The most predominant type are these arbuscular mycorrhizas that make up about 78% of all plants. They're ubiquitous among grasses and forbs, and many tree species are arbuscular mycorrhiza. And it's important to know that these mycorrhizal relationships are fixed based upon the species identity of the tree or plant you're looking at. So if you're looking at, say, a sugar maple, you know it's going to form arbuscular mycorrhizas because they're very context and species dependent. I am personally interested in this other group of mycorrhizas we have called the ectomycorrhizas. And while they only represent 2% of plant diversity globally, they're incredibly important in forests because a large portion of our trees that we study form these ectomycorrhizal symbioses. And in fact, it's almost an even split in the relative abundances of ectomycorrhizal trees and arbuscular mycorrhizal trees in any given forest that you happen to go to. So if you were to go, say, hike around Baker Woodlot, you'd be equally likely to encounter a nice ectomycorrhizal beech as you would a red maple, which is an AM tree. So despite their underrepresentation from a diversity standpoint, they're really abundant in the forests that we care about. However, they do have very stark differences, both physiologically, functionally, and morphologically, that end up setting the stage for generating these different feedback mechanisms we're going to talk about here in a minute. So the ectomycorrhizal symbiosis primarily grows around the outside of the tree's root system. It has this inner Hartig net that actually snakes around the cells just under the surface, and this is where that exchange of carbon and resources take place. And because of this, these fungi are able to really grow off of these root systems. They're able to really explore the soil environment. And you can actually look at them under just a simple dissecting microscope, count tip numbers, and figure out how colonized your root systems are. Arbuscular mycorrhizas, on the other hand, are a lot more discrete. They don't grow nearly to the extent that ectomycorrhizal fungi do. Their mycelium actually penetrates inside the plant cells themselves, which is where the physiological exchange of those nutrients and carbon takes place. And then they don't really produce a lot of biomass on the outside, and they don't cover the outside of the root. In order to visualize these, we have to use some fairly straightforward staining techniques where we bleach the roots and then stain them with this nice blue dye. And then we can observe the fungi under a compound microscope instead of a dissecting microscope. So they're a lot smaller. Why we should care about mycorrhizal associations out being foresters, besides the fact that they're cool, is the fact that in addition to species composition changes that are being caused by climate, things like deposition, selective logging, we're also seeing shifts in what we call mycorrhizal dominance. And kind of put simply, this is the idea that in many forests here in the eastern United States and globally, ectomycorrhizal trees are dying and they're being replaced by arbuscular mycorrhizal trees, almost in fairly straightforward directions. Here in the south, it's a little bit different, but mostly up north, especially where we're at in Michigan, we're seeing a lot of this AM dominant systems replacing what were historically ecto-dominated systems there. So we see, despite these species composition shifts, which if it's one species replacing another, we can't really make predictable trends. We can make some fairly substantial hypotheses and claims when it's entire groups of these symbiotic organisms changing regardless of really the species identity of the trees there. So this is where this idea of plant-soil feedback comes into play. And we think we can use plant-soil feedback based on mycorrhizal types to help explain these community dynamics and figure out where they're going in the future. So plant-soil feedback outcomes come in two flavors. The first type of feedback we have is positive plant-soil feedback. This is primarily driven by mutualists like mycorrhizal fungi that grow in the soil system. And as any given population of plants grow, they reproduce, their offspring are recruited. That grows the populations of the mutualists in the soil, which then promotes the growth of their host plants, which then further promotes the growth of the mutualists, which further promotes the growth of host plants and their offspring recruitment. So there's this positive feedback loop that occurs with these plants in their soil environment, and this can lead to outcomes where one plant species becomes more competitively dominant than others and can outcompete others in the system, leading to either monodominance or the competitive exclusion of specific species in the population or the community. On the other hand, negative feedback results in the opposite outcome and is primarily driven by interactions between plants and pathogens. And in a similar style of feedback, as a plant grows and recruits its offspring, host-specific enemies and those pathogens in the soil also grow, which then dampen and retard the growth of those plants themselves. So there's this ebb and flow, and as the plants grow bigger, their pathogens grow bigger, which then causes die-offs in the population, which then causes the dampening off of the pathogens themselves in an ebb and flow type relationship there. And while that's not necessarily maybe a good thing for any particular species trying to gain a competitive advantage, where this results in a community is this sequential turnover of who is dominant and who is not. And negative feedback outcomes can actually help promote plant diversity. So the fact that they're negative isn't really necessarily a bad thing if we care about how to maintain plant diversity versus monocultures or monodominance there. So these cyclical patterns of growth, shrinking, and swelling of the mutualists and pathogens in the system leads to an observation known as conspecific density dependence. And this built off of some older literature and work done by Jansen and Connell in the 1970s. So if you've heard of the Jansen-Connell hypothesis, this is kind of the logical extension and the burgeoning of that field there. But this is the idea that essentially, if we have these feedback relationships going on in the soil environment, we should be able to observe this with the relationship between plants and their offspring. And in the case of a negative feedback scenario here, this soil environment should be engineered with pathogens, they're growing, and essentially what ends up happening is you get fewer seedlings and juveniles being recruited nearby adults. And under positive feedback, what we end up seeing is conspecific positive density dependence. And as the density of adult trees or plants increases because of that positive benefit from their mutualists, you also see increased recruitment of those offspring in that system. So we can get two fairly different dynamics and spatially dependent outcomes based upon these feedback processes. So visualized another way, I'm going to throw some mathematical graphs at you, but we've tried to keep them simple when we publish this paper. It's just a straight line, no fancy equations to it, just y equals mx plus b. Here on the y-axis, we have a proxy for fitness. This could be plant growth. Here we've defined it as offspring success. And then on the x-axis, we have conspecific adult density. Now, historically, plant soil feedback work has identified that plant soil feedback typically results in a negative trend between any given plant and its population size. This is primarily driven by the fact that host-specific pathogens tend to build up faster than mutualists do, and they have a tendency to override the mutualists in the soil. And that leads to a negative trend. As you get increasing conspecific densities, you get lower offspring success. But most of us in this room are ecologists, right? We know that a single straight line doesn't really explain anything in ecology because variation in the system exists. So really, this is a much more accurate representation of plant soil feedback outcomes across all plants. You have some that are experiencing very high dampening off, very high negative feedback, and then some that are experiencing that positive feedback, which can tip the scales in favor of conspecific positive density dependence. So that's all well and good, but usually these cases are stochastic, they're species specific, and it's really hard to define obvious trends as it relates to feedback in the plant world. But in 2017, a landmark study came out from Jonathan Bennett's group that grew one generation of different trees on soil and then replaced that generation with a second generation of the same species. And in a plant soil feedback framework, if positive feedback is promoted, you see higher growth in that second generation. And if negative feedback is promoted, you see worse growth in the second generation compared to the first. And when they looked at all their tree species, they sorted them by mycorrhizal type, and what they found was that for the most part, ectomycorrhizal tree species grew better in the second generation and were promoting positive feedback, whereas in the arbuscular mycorrhizal trees, with the exception of a couple of weirdos up there, they were largely generating negative feedback with their second generation growing worse. So I have never seen data look this good ever, and it's incredible. So this was a big deal back in 2017 and really still is now. And it was their belief and our belief as we were working through how this could be generated on the basis of mycorrhizal types, that it probably relates to those core physiological, morphological, and physical characteristics that differ between mycorrhizal types, and then other traits that seem to be related to these mycorrhizal syndromes, such as the recalcitrance of the host plant leaf litter. And in the case of ecto trees, they're pretty recalcitrant. They have really low C to N ratios. They have a lot of tannins and lignin, and they're really hard to just decompose. Whereas arbuscular trees primarily have fairly labile leaf litter that's real sugary. It's got really simple carbons, and it decomposes very quickly. So this is one example of a trait that is largely indicative of these mycorrhizal syndromes. So if we're going to build on our conceptual model here and complicate things like us ecologists like to do, we can think about how a generic plant, if it's ecto or arbuscular, can change its feedback outcomes and change its density dependence based upon that prior work, where ecto plants experience positive to neutral feedback, generating neutral to positive density dependence, and AMs experiencing negative feedback. And in the last 5 years or so, there have been many studies that have shown this effect of conspecific density for ecto and AM plants, specifically in a wide variety of forest systems, temperate hardwoods, tropical, subtropical, boreal, so on and so forth there. So we think there are 3 ways that mycorrhizal associations can affect plant soil feedback. And this primarily has to do with interactions between free-living saprotrophs in the soil, interactions with pathogens in the soil, and in the strength of the mutualistic interactions themselves. So several of these next slides are going to look at some form of this where we're going to compare and contrast what's going on in an AM system to an ecto system. There's a lot of art on this, and we'll walk through them very slowly. So we're going to spend some time getting to know these different systems here. Arbuscular mycorrhizal fungi, indicated by these little guys here, are incredibly good at taking up mineral forms of nutrients like nitrate, phosphate, and ammonium from the soil environment, in addition to water. However, these resources don't just freely exist in the soil system, and they have to be converted into these mineralized forms from leaf litter and soil organic matter by a free-living saprotroph community. These saprotrophs are heterotrophic like you or I do. They are chewing through this organic carbon with their enzymes in order to respire it so they can get energy and grow a little bit. And because they're carbon limited instead of being nitrogen or phosphorus limited, they're secreting all of that as mineralized waste. So these mycorrhizal fungi then gobble it up, they get carbon from their host, and everybody's happy. This relationship is actually so important that arbuscular mycorrhizal plants will actually exude small amounts of carbon from their root systems in order to prime the microbial community and really encourage rapid decomposition of the soil organic matter around those root systems so they can get advantageous benefits from that. However, this comes with a couple of different consequences to it. The major one that I'll talk about is this fact that some saprotrophs are capable of acting as facultative plant pathogens. If you really think of what like a soil organic matter residue might be or what leaf litter is, right, it's senesced plant parts and they're full of cellulose. And you need cellulase enzymes if you're a saprotroph in order to decompose that cellulose. Well, living plants also have cellulose in their plant walls. So if their immune system is not that great or food is fairly limited for these saprotrophs, many of them can switch and use their enzymes in order to attack plants in antagonistic ways. So what is a good relationship between the plants and the saprotrophs in order to liberate nutrients can actually promote negative feedback by encouraging the growth of these secondary facultative saprotrophic plant pathogens. The ectomycorrhizal side of the equation here is a little bit more interesting. This is really dynamic, but what I think is really cool about these ectofungi is numerous times and through evolutionary history, different lineages of ectomycorrhizal fungi have evolved from saprotrophs themselves, and many have retained that enzymatic capability of decomposing and interacting with soil organic matter and leaf litter. And what this results in is a situation where instead of being reliant on a community of saprotrophs to liberate these nutrients, these ectomycorrhizal fungi can actually go and get that organic forms of nutrient directly through their own decomposition and donate it to their host trees so that way they can get carbon from that. So this creates a scenario where instead of facilitating and priming the growth of saprotrophs, these ectomycorrhizal fungi actually outcompete and competitively exclude these saprotrophs in the soil. This is known as the Gadgil effect. So this can generate positive feedback instead of negative feedback in two ways. First, there's no facultative saprotrophs or facultative plant pathogens hanging out because they've competitively suppressed them. And these ectomycorrhizal fungi are giving their plants unique access to this really nice resource pool that nothing else is capable of accessing. So it creates a really tight feedback between the plants, their leaf litter, and their mutualists. Moving into a little bit more straightforward territory, we have direct interactions with pathogens. I will stick on that ecto side because it's a little bit more straightforward. I mentioned earlier they have that physical barrier around the root system, and this is hypothesized and believed to provide a physical barrier for pathogen infection that just prevents pathogens from interacting with the root systems to the extent that AM fungi and their host trees might be subjected to. So that's great. There's still pathogens in every system, right? We can't get germ-free, but this probably leads to very weak levels of negative feedback being generated between ectomycorrhizal trees and these pathogens in the soil. On the other side of the equation, the arbuscular mycorrhizal trees and plants, they don't have it quite so good. We don't have that physical barrier to protection, so pathogens are a little bit more able to enter those systems and attack these trees. The added mineral nutrient nitrogen in the soil can increase tissue nitrogen content, which has been shown to also exacerbate plant disease and encourage the growth of pathogens because the tissue is just a little bit more chemically rich and diverse and they want to eat that more. And then lastly, because the arbuscular symbiosis has to actually grow into the cells of the plants themselves, like they're fungi and the pathogens are also fungi. So they, in a lot of ways, have to communicate with the plant and tell it to ignore signs of fungal infection because, yeah, they're infecting the plant, but they're doing it for a good physiological reason. So oftentimes the plants will downregulate their immune system in order to form the AM symbiosis, which can make them vulnerable to pathogens. So this can promote pretty strong negative feedback. The last area where we see some differences between these systems is in the strength of their mycorrhizal network interactions. There's currently a lot of controversy in the field of mycorrhizal networks. There's been some suggestion that they can communicate, like trees can communicate and pass nutrients back and forth. That is not supported in the literature. And we're still doing a lot of research to see to what degree that could potentially take place, if at all. But one thing we do know about these mycorrhizal networks is they do connect trees. And in the case of ectomycorrhizal fungi, they have that really big mycelium that you can think back to that first slide growing everywhere. And if you have a dense mat of that growing through the forest, a seedling germinating and entering the system for the first time can potentially tap into that network and start forming that really beneficial symbiosis fairly quickly, as opposed to having to grow and invest all of the carbon for themselves. So in a way, the adult trees subsidize the carbon cost for the formation of these critical mutualisms for their seedlings. So the more adults you have, the more mycelium you have, the better environment it is for your seedlings there. Arbuscular mycorrhizal fungi don't help out quite as much. Again, they don't have much biomass. Their mycelium is not very big. It doesn't go very far from the root systems, and it turns over relatively quickly, which requires a consistent investment of carbon in order to maintain those. So there's not a lot of the mycelium for seedlings to tap into. And there's also the problem that when mineral nutrients are really highly abundant, like say during times of strong indeposition or fertilization, plants can get all of the nutrients they need through passive uptake when they take up water from the soil. They actually don't need their mycorrhizal symbionts in some cases in order to get all of that nutrition. But the mycorrhizal fungi are still hanging out and they're still taking carbon from their host plants. So when resources are fairly rich, this mutualism can actually slip into parasitism territory and become a net negative instead of a net positive benefit for the mycorrhizal fungi themselves. So again, this is going to promote slightly weaker positive feedback compared to the ecto side. So taken together, we have two mechanisms in AM plants that promote really strong negative feedback and one that promotes weak positive feedback, which can explain this outcome in the AM plants. And then on the ecto side, we have two mechanisms that generate very strong positive feedback and one mechanism that helps tamp down some of that negative feedback generation, explaining that trend. So we know there's a huge microbial component here at play, how trees interact with the other microbes that aren't just their mycorrhizal fungi. So what does that mean for our scenario where we're losing ectotrees and they're becoming more AM dominant? Now we can start thinking about like, well, what's going to happen with these underlying soil communities in this hidden half that these papers don't necessarily address? Okay, everybody's an expert now. I love it. Let's move on to the actual data and the actual work that I've done besides making those really nice cartoons. So again, we now know that there should be these hypothesized interactions with these microbes in these soil systems that are different. So can these tree and microbial community dynamics affect other trees and soil in the system through these dominant spillover effects that occur? So to look for evidence of this, we're going to look at some work that I've done examining forest soil microbial communities with next generation sequencing. For these projects, we extracted DNA from the soils, looked at the fungal communities and what they were capable of doing in some fairly slick experimental designs out in some forests. And we're primarily going to be looking at the relative abundances of saprotrophs, pathogens, and mycorrhizal fungi. So I'm not going to get into the nitty-gritty taxonomy. We're going to stay at this nice functional group level for our levels of understanding. We're also going to look at the diversity within these groups. And then the last bit of data that we're going to look at is seeing if the microbes we're seeing and these feedback drivers actually contribute to changes in forest spatial structure and the relationships between adult and juvenile trees through density-dependent interactions. So this leads us to this mycorrhizal spillover hypothesis we've been working under. This is the idea that these dominants, like the collective mycorrhizal type of a dominant species, can engineer the soil environment in ways that spill over to less abundant plants, regardless of their species identity or their individual mycorrhizal association. And under this hypothesis, we expect to find more pathogens and more saprotrophs in AM-dominated forests, and therefore more negative plant soil feedback and negative conspecific density dependence in these systems as well. For you that really like to dig into your old ecological theory here, this is consistent with the idea of foundation species that Ellison has been working on for a while, or the mass ratio hypothesis, which essentially states that ecosystem functioning at any given point in time is primarily determined by trait values of the dominant contributors to plant biomass. So whatever plants are most abundant on the landscape, they're going to have the biggest effects on soil and everything else. It makes logical sense, right? So to explore these, I've done work in 3 different states. I'm going to share with you a couple of projects from South Central Indiana and upstate New York first, and then we're going to go halfway here to Ohio, unfortunately Ohio, and talk about some spatial evidence that I've looked at there. So in South Central Indiana, this is where Rich Phillips works, and he's going to talk to you about many of these plots as well. We have several forests that have their tree communities studied, and we know the mycorrhizal types of all these trees, And these plots range in their mycorrhizal dominance from 100% arbuscular mycorrhizal trees to 100% ectomycorrhizal trees to plots that are 60% ecto, 40% arbuscular, 55-45, and so on. It's a really neat trait when the way we're able to quantify these communities based upon these basal areas on a continuous metric. So we sequenced all of the fungi for this, and I want to show you just two plots that represent like 4 years worth of work on this, which it kind of feels bad, but it shows a lot of data. So first, we'll orient you to the x-axis here. This is on a gradient of AM tree percent relative basal area. So points towards the 100% side are really heavily AM dominant plots. Points towards the 0 side are really ectomycorrhizal dominant plots, right? 0% AM tree basal area means there's 100% ecto tree basal area there. On the first y-axis we have on the left-hand side of the figure, we have just our fungal relative abundances, and then the right-hand side is the taxonomic richness of these two groups of fungi. The colors correspond to soil saprotrophs in the green, plant pathogens in the red, and then the part that you can't really make out is the differences between richness and abundance, but that doesn't matter because if you pay attention to the trend lines, everybody's increasing with increased AM tree relative basal area. So our AM dominant plots here in Indiana have higher diversities of soil saprotrophs and plant pathogens, and they have higher relative abundances of saprotrophs and plant pathogens in their soil. So this is a cool finding. Another cool finding, but one that's a little bit more obvious, is when we looked at the ectomycorrhizal fungi. As you increase AM tree basal area and you lose ectomycorrhizal trees, you also find less ectomycorrhizal fungi and lower diversities of ectomycorrhizal fungi. This was probably the least surprising thing we found overall, but it's also good to have confirmatory results like this because they can show us that we can really rely on those other trends where we are seeing these increases and they're not just spurious correlations there. Okay, so at least in Indiana, we know these AM communities have more saprotrophs and more pathogens, and they're more diverse. But how well does this hold up in other areas, and can we potentially look at the effect of environmental context on these interactions? In our Adirondack plots, we had 3 different locations. They spanned a gradient of being in a warm, dry site off to the eastern side of the range, a cold, wet site being more to the western side of the range, and then an intermediate site that had roughly the same precipitation regime as the cold site and the same temperature regime as the warm site. So kind of a nice middle ground there. For these plots, we specifically, instead of just setting up regular plots, we anchored them around a central focal tree of ecto or arbuscular mycorrhizal type, one that was pretty big and had obviously been there for a long time. And then we censused the community around those trees and quantified its mycorrhizal dominance. So with all of the different points we have across all of the 3 sites, we have arbuscular and ectofocal trees that are in AM and ecto-dominant communities and some gradient in between. So we're able to sample locally around the tree and see what the contributions of the surrounding community are on the fungi. What we found here has been pretty cool as well. I'm just going to show you a little bit of data for the plant pathogens. These x-axes are reversed from the previous study since this is a more heavily ecto-dominated region. The people reading these types of papers care a little bit more about ecto trees than AM trees, so we presented it that way. But what we have towards the left-hand side of the plots are our more arbuscular dominated communities, and at the end of the plots on the right-hand side are the really heavily ecto-dominant communities, and then the gold corresponds to focal trees that are arbuscular, and the green are ectomycorrhizal focal trees. And what's super cool, I think, about this data is if we think about what's going on with the AM trees and our ecto plots here, they're encountering really relatively low abundances of pathogens and low pathogen diversity. However, as the community around the AM trees becomes more AM dominant, we get this very clear increase in the diversity and relative abundance of these plant pathogens. So as the AM community increases in dominance, the trees in those communities are going to be encountering more and more pathogens. Our ectomycorrhizal focal trees, when we sampled around them, they seemed largely insulated from this effect, which probably lends some good credibility towards that hypothesis that maybe they're more better defended, and as a result, we have less localized pathogens around those big focal ectomycorrhizal trees. So that's cool. We have interacting effects of these individual trees and surrounding communities. We have spillover occurring of the dominance of these mycorrhizal types. We also found that the strength of these interactions can be context dependent. We saw the strongest trends in relative abundance with our warmer and drier site, whereas our cooler and wetter site largely didn't show a lot of those trends. And as we know in the Adirondacks, right, it's a very cold and wet region anyway. So our coldest and wettest site here was likely having some superseding effect of climate or geography overriding a lot of these nitty-gritty interactions and just slowing the entire system down there. But that's really cool because there's obviously a huge role this can play in global climate change. Okay, so if we're building on our model here, this was our initial model split out by plant mycorrhizal type. We know, according to the Bennett paper, ectoplants positive to neutral, AM plants negative. But now we can think about what happens to a generic plant in an ecto and an arbuscular community. And what we likely see is if you're just any given plant, pick your favorite one, you're likely going to perform better in an ecto-dominated community than an AM-dominant community because there's fewer pathogens, fewer saprotrophs, and maybe more of those mutualists if you're also of the same mycorrhizal type. So that's cool. We got this really compelling microbial evidence, but obviously we just looked at DNA in the soil, right? Is this actually affecting tree communities? So to look at that, we did a spatial analysis of a property owned by Kent State known as Jennings Woods in Northeast Ohio. Over the course of a summer, I and a whole team stem mapped spatially about 8,000 different trees, and based upon their size, we classified them as being adult trees or juveniles based upon a cutoff of their basal area. And you can see here, we have a nice blend in these plots of mini ecto, mini arbuscular, and a nice mix of both. So in total, we censused about 28 plots with this, and I think it's really neat. These plots are only 15 meter in radius, and you can see several scenarios where if you just go 10, 15 meters away from the center of a plot, the mycorrhizal dominance of these plots changes very rapidly. So we're really testing for community level spatial effects that are turning over on fairly reasonable scales for us to observe here. And to test this, we did what's called a point pattern analysis, where you can take points of adult trees, leave them fixed in place, randomize the juvenile trees' spatial location relative to the adults, and then compare that spatial pattern of randomness to the observed patterns that you've actually censused with your data, and you can try to figure out if your trees are more clustered or they're spreading further apart, given just a random chance where they dispersed into one another. So our findings were pretty cool. We found that there are fewer juvenile trees living near adult trees in AM stands. And this didn't matter if it was an ecto tree in an AM stand or an arbuscular tree in an AM stand. And you can see that here on the two graphs that are on the left-hand side. We have this distance from a juvenile to an adult here. And then the y-axis is the density of those individuals. And there are lower densities at shorter distances nearby adult trees, and as you move further away from the adults, you get higher densities of these adult trees, indicating that if everything was uniform and dispersal, something is making that environment near the adult trees antagonistic. It's the pathogens. Meanwhile, if you move over to the ectomycorrhizal side of our forests, here we see that trend tip in the opposite direction, and in our most heavily ecto-dominant plots, here we go, right here, we have higher densities of juvenile trees at closer distances to adults and fewer juvenile trees further away. So they're trying to recruit more of those juveniles that are close by. We also found that this stood for the heterospecific and the conspecific ratio of these, where AM dominant plots had higher proportions of heterospecifics versus conspecifics, indicating that the more dominant species were suppressing their own offspring to a greater extent. And in our ecto-dominant plots, we have much lower ratios here, so we have much higher recruitment of conspecifics around those really dominant species there, which is another maybe more or less kind of clear sign that negative density dependence and positive density dependence are splitting off by the basis of mycorrhizal type. So kind of putting a much better graphic that's not actual data, we can think about this from our modified figure, where we have this AM dominant community resulting in recruitment of juveniles further away, in addition to lower conspecific recruitment. And in the ecto side of things, we have higher recruitment of juveniles overall at close distances and increased recruitment of conspecific individuals there. So while we were thinking about this idea of spillover, some other researchers were thinking about probably a more straightforward idea, and that is if a given tree just has more of its same mycorrhizal type individuals around it, regardless of their species, can that just give a net benefit to these trees? Hopefully they'll just have more mutualists around them. And they looked at the change in per capita sapling density surrounding conspecific adults and showed that AM and ecto trees globally have lower densities of conspecifics surrounding their adults. But when the adults were all aggregated based upon con and dissimilar mycorrhizal types, there was a positive effect seen. So they pitched this as this con mycorrhizal density dependence, and as you have more con mycorrhizal individuals, your offspring have a higher success at establishing. So kind of put on our nice hypothetical diagrams here, what this con-specific mycorrhizal density dependence looks like for a generic plant means that if you are in a community of your same mycorrhizal type, maybe your offspring just see inherently higher success, but you're still experiencing this negative density dependence. Whereas if you're a plant in an different mycorrhizal type community, the heteromycorrhizal communities, you have a lower offspring success in general, and you still experience that negative density. So one of the things we really sought out to do here,
we don't know when the furniture could be delivered yet.
Correct.
They have a text.
some of the stuff that we intended to do here was try to unify the two interactive and additive spillover models into this complex diagram that has a lot of lines on it. Essentially what we see here is that in ectoplants and ecto communities have the intrinsically high offspring success because of the mycorrhizal type match, and because they're in a community of ectomycorrhizal plants, they're getting this positive density dependence through positive plant soil feedback. Meanwhile, AM plants and AM communities likely have pretty severe negative feedback leading to negative density dependence, but maybe their offspring just have intrinsically higher success because they have plenty of those extra mutualists around that they need to care about. Meanwhile, in these mixed mycorrhizal type scenarios, an AM plant and an ecto community might have lower offspring success, but experience not quite as severe negative feedback and negative density dependence because the ectomycorrhizal contributions from the community are swamping out a lot of those pathogens and those saprotrophs that can be antagonistic. And maybe the worst case scenario is if you're a single ectomycorrhizal tree in a big community of AM plants, right? You're a seedling dispersing into this environment. Maybe your offspring success is inherently lower because there are fewer mutualists to associate with around, and then you experience fairly strong negative density dependence and negative feedback because of the superseding of abundance of pathogens and saprotrophs in the system here. So knowing we have these complex plant microbe interactions that split upon mycorrhizal type, we know that the community composition of these mycorrhizal types is important, their dominance is important, and we know it can actually affect recruitment and the spatial structure. Where do we go from here, right? What are the consequences of these spillover effects on global climate change itself? I promise this is the last time we're going to look at this figure. What we think why these spillover effects matter is while we're getting these shifts in composition, we may be getting them for an additional reason, and that could be related to these feedback processes where AM trees have an easier time expanding into ectoforests because there's fewer pathogens and fewer saprotrophs there. So they're able to essentially undergo some form of enemy escape to initially get established. This also means these are likely difficult to tip back the other way because as ecto trees try to repopulate and reestablish in more heavily AM dominant communities, they're going to be contending with fewer mutualists and more pathogens and saprotrophs that their mycorrhizal fungi have to compete with and defend against. So it may be, we're seeing these changes and they're going to be reinforced by these plant microbe interactions. This also has kind of bigger consequences from a holistic standpoint, and there's been several papers that are showing this. As you get more AM trees, right, we're expecting to get more pathogens there, and these studies have all shown pretty much this horrible red figure, that globally, the relative abundance of pathogens in pretty much every ecosystem is going to drastically increase. And this is due to numerous factors unrelated to plant mycorrhizal type, but I hopefully have convinced you all that it may be because of plant mycorrhizal type as well. At least it's not helping there. Thinking even longer term, we're going to get into some interesting evolutionary space here. These changes in these soil microbial communities should have fairly significant consequences for the change in selective pressures from nutrient limited and nutrient acquisitive systems towards selection being primarily driven by plant pathogen interactions. And in this case, we might see some form of promotion of diversity, whether it's species diversity or genetic diversity. And as we get higher diversities of pathogens and we have more selective pressure from pathogens, maybe we're also seeing increased genetic diversity in populations that need to better defend themselves in order to have higher fitness. I think this is going to be very challenging to study going forward because trees are long-lived, they produce a lot of offspring that all die, and these dynamics don't really operate on timescales of like small grants to fund and study, or really human lifespans either. We need like a couple hundred years of monitoring the same set of forests in order to do this. But it's an interesting future direction to kind of think about that adds this added layer of like what happens if the world just becomes more pathogen dominated in these ecological systems. So we've covered a lot through this talk, but hopefully I've convinced you of a few things, and I'll leave you with some take-home messages that should help kind of pull key pieces of information from this. So we know now that tree mycorrhizal associations can generate different plant-soil feedback outcomes through these different interactions with the free-living microbial communities in these soils. AM tree species are more prone to negative feedback. Ecto tree species are more prone to positive feedback. These interactions can definitely affect trees and soil microbial community dynamics through these mycorrhizal spillover effects and through the mass ratio hypothesis. We see that AM dominant communities are more prone to conspecific negative density dependence because they have more pathogens and saprotrophs, whereas these ectosystems are typically more prone to more positive density dependence because they have less pathogens and more, in some cases, beneficial mutualists for other plants that are growing there. And we know that these spillover effects should and will have consequences for forests under global change. We know that negative feedback and positive feedback are going to result in conspecific negative density dependent drivers being promoted, while positive density dependent drivers are going to be diminished. We also know there's going to be more, hopefully, maybe, we're predicting, more selective pressure from pathogens and less selective pressure from mutualists and resource acquisition, and more diversity from both a genetic and species standpoint. So with that, that wraps everything up. Again, I want to thank everybody that I've worked with, and I'd be happy to take any questions.
Great presentation.
Thanks, Aki.
And the questions?
So when you mentioned about pathogens and sacrifice in your feedback model,
Yep.
what pathogens were you considering? Because in forestry system, oftentimes you have pathogens, which will normally can be also be sacrificed
Yep.
under the suitable circumstances. So how do you differentiate those in your model?
Right, yeah. So the question was if we can distinguish the effects of pure saprotrophs from pathogenic fungi and then those that are capable of doing a dual role. I believe that's what you were asking, right? Yeah, I am working on exploring that in the data sets that we have because it's a really compelling hypothesis about this duality of the roles. Through the tools that we have as fungal ecologists, there's a great database that's been curated out there that has primary and secondary roles for a lot of the well-known fungi that may be detected through sequencing studies. And in the cases where I'm looking at the saprotrophs and the pathogens, I'm just working on one of the functional categories. It's whatever their primary function is, whatever they have been in the literature documented doing most of the time. I have not dipped into that secondary category, and I haven't pulled specifically the fungi that have maybe a pathogen listing and a saprotroph listing in either category. But I am working on pulling that, and that's kind of brings up a good broader point to acknowledge about this type of sequencing work. You know, I consider myself, I wear a lot of hats, as you can see from the presentation, microbial ecology, forest ecology. But broadly, I think I would classify myself as a community ecologist, right? So most of these data sets are at a really high level where we're trying to look at these really generalizable trends among these groups. I have not done any work with all of the data sets we have, really exploring too much of the taxonomy, so who the individual fungal players are with these. We do have a joint experiment that I'm working on data for now where we've done an ingrowth core experiment in each of these forest locations. So we have a steady temporal sampling that we're hopefully going to be able to identify like which pathogenic fungi may be regionally dependent on the 3 states, for example, or which pathogens may be driving these trends more ubiquitously, and hopefully be able to point to like an actual fungus name and start telling people about that. So it's one of the next phases of this.
Just a follow-up.
Yeah.
On that. So in your model, you included mycorrhizal, saprophytes, and pathogens, right? And then where do you see rhizobacteria, which is also the integral component of our soil?
Right.
It stands there. So did you include any of that in your model? Because as it control nutrient cycling
Sure.
and competition against pathogens
Mhm.
and saprophytes, or maybe with mycorrhizal?
Yeah, so there has been one paper that has also done the bacterial sequencing. It's Baram et al. 2020, Baram et al. 2021. They looked at like fungal bacterial ratios. I don't work with bacteria. One, I may make some people mad with this. I consider fungi to be more important in ecosystems, both from a biomass standpoint and just from their functional capabilities and what they're able to do. And from the literature that I've read and the data that I've seen, they are more important than bacteria. Like, not to say bacteria aren't important, they don't contribute to these things, but it's predominantly interactions with fungi that are driving these trends. There's also, in my experience of working with bacterial sequence data, we don't have nearly as good of a functional database as we do for the fungi. Like, we can take fungal taxonomy and slap their functional roles on with a really quick command at R, because the database is just an Excel sheet you download. The databases that I've worked with for bacteria, a lot of them are very like gut microbiome and human health focused, and there really hasn't been a concerted effort from, like, say, soil bacteriologists to put together that type of functional capability database in that way. So the bacterial communities I've screened with my sequence data, I'll get all sorts of crazy stuff like the number of bacteria associated with Alzheimer's diagnoses. And I'm like, I'm looking at a dirt sample. That is like not relevant. So it's been too much for me to parse, but I think that is a good future direction to go into as well. Sorry, the question was, what role do some of the bacteria play in these systems too, if that wasn't clear? Yeah.
Nice talk. That was
Thank you.
really interesting. Very cool work. And I have to commend you too for not just, culturing soil and working in the greenhouse, but doing the actual spatial patterning.
Thank you.
So I had just kind of a couple of questions on that. I think it was in Ohio where you were looking at the spatial pattern.
Yeah.
One is, so what did you consider a juvenile?
Right.
And like, because you have everything from something that might be a first year seedling, which may have or may not have a future, to, decades old saplings. So that one question, and then the second was, what about shade tolerance? That seems like the shade tolerance of the species in question could be potentially confounding in interpreting these patterns. And is that something you considered or thought about in this work?
Sure. Yeah. So the question was a two-parter. One was a methodology question about how we classified our adult and juvenile trees here in this study, and I'll leave this slide up. It's a little bit more interactive. And then the other one was about the species shade tolerance abilities and kind of these species-specific effects. So both great questions. Yeah, so from the methodological side, we looked through the literature as far as like how tree ages are estimated based upon DBH. And there really wasn't a consistent through line. Each study just seemed to kind of like arbitrarily define it, which was really unsatisfying for us and meant we also had to arbitrarily define it and then argue our case for it. So we considered anything. So first of all, we censused everything that was diameter at breast height and above.
For both juvenile and adult?
Well, yeah, for all trees. We did everything that was DBH height and above. And well, that's not true. We did everything that was greater than 10 centimeters and above. And then anything that was not of a DBH height, we recorded its height. And those were all considered to be juveniles, right? And then anything that was of DBH height and above, if it was between 0 and 5 centimeters, we considered that to be a juvenile. And then anything 5 centimeters and above, up to like 50, 80 centimeter DBH, was considered to be an adult. So we do have this gray area between, like the true saplings and then some of the trees that are about that big, where we're not really sure like how old that tree is, but we are reasonably confident that the majority of the data are bigger trees in these plots. and they would be considered to be much older than the saplings being recruited in. We tried to do seedlings and everything 10 centimeters below in this, but it took us a week to do an entire plot because there were just so many seedlings, and it was not reasonable to do from a spatial scale. So I did that on a much smaller area, and it didn't end up working out because we shrunk the plots we were doing to like 5 meters, and there wasn't enough adult tree presence in order to have the statistical power to do the point pattern analyses with one of the 2 points. Second question about shade tolerance. I really like the literature on shade tolerance. I'm not sure if you're familiar with Sarah McCarthy Newman. She's done a lot of feedback research as it relates to shade tolerance and how shade sensitive versus insensitive experience different strengths of plant soil feedback. I think it's great work. I've tried to take a higher level than that because I'm fundamentally on this border between community and ecosystem ecology. And I really try to relate kind of these outcomes to big ecosystem level effects in that regard. So in my case, I'm not necessarily interested in what any particular species is maybe capable of doing or how resistant it is. I just want to try to find generalizable patterns as I group these species together. So we weren't doing any work with the shade tolerance of those. But if you are interested in how that can potentially fit in, Sarah Newman is doing a lot of work on the mycorrhizal spectrum as it relates to feedback with shaded experiments, non-shaded experiments, and things like that. And it does play a big role.
I guess I was thinking about it more from this spatial pattern perspective, in that
right.
Some of your, I would assume some of your ecto species like oaks, you may be very challenged to find juveniles
yes.
at all because of their shade tolerance, whereas a peach, maybe so. And then I would assume, I'm not sure what, I assume you have a lot of maples.
Yep, a lot of maples.
Dominate your arbuscular, which are going to be shade tolerant.
Yep.
And they're going to be ubiquitous.
Yeah.
So I was thinking of it more from this perspective of
definitely
the density, evidence for density dependence in the field.
Yeah, so one of the cool things about this functional ecology paper is we did generalize based upon these species, right? We lumped them all. Jennings Woods has about 30 different tree species. You know, maple is dominant, a lot of red oak is dominant, beech is very heavily dominant, but we have a couple different hickory species. We have some liriodendron out there. You know, it is the secondary ranks. We used to have a lot of white ash, yeah, but we have camera resolution data showing that the immediate canopy that we were sampling was all roughly more or less the same density there. And we were hoping, we did all these analyses on the ones where we had the power to do it, the most abundant ones, on the species level as well. And we were able to actually show really cool outcomes. Like with red oak, we had red oak that would have juveniles recruited around it in the ectomycorrhizal dominant communities, but we would have adult red oak individuals in these AM dominant communities that had no juveniles around them at all. So we took that as a sign of like, okay, well, they can clearly establish and grow in these forests regardless of minor shade differences. We're just not seeing any recruitment of oaks in the arbuscular mycorrhizal plots, despite the fact that the adults are at low density, so you would expect density dependent to be really favorable to those interactions. I wish we had more data for this. Again, this was kind of a small forest, and this was a huge effort to stem map. You can imagine mapping and trying to get the spatial location for all these trees. And over the course of a dissertation, we just had one summer. I would love to set up much more intentional plots where things are mapped and monitored for a lot longer time, and we have a big enough sample size at the species level resolution. That's the same thing with the Adirondacks design. We don't have the species identity replicated in the plots. We'd have plenty of ectos and plenty of arbusculars, but I think sugar maple is the only species where we have like more than 5 focal trees in both ecto and AM dominated plots. So, and of course, if we just look at one species, we can't really generalize. So, but good questions, thank you. Were there any questions in the chat?
Not yet.
Not yet, that's fine. Yeah.
If we have time.
Yeah, one more question.
You're holding me up for lunch, Jeff.
So you've done a great job communicating to, I assume, a number of people that are new to this area of work. Evidence for a mechanism that may operate at, you know, the neighborhood scale at a specific point in time, and the development of sandwiches, seedling recruitment.
Right.
I'm wondering if you go back to this trend in changes in mycorrhizal abundance, if you can think of any other types of mechanisms that operate at larger spatial scales or across larger temporal scales, and I'm specifically thinking of disturbances like fire or insect outbreaks, and of course management that might contribute to, for example, decline in ectomycorrhizal species in the northern part of the U.S. due to lack of fire and less intensive management, or the increase of the importance of pines in the south, and if maybe there's other things that could be operating kind of at larger scale that are also important in determining kind of the diversity and composition of forests.
Yeah, most certainly. So Jeff's question was about, like, this is a very big geographic extent, right? You have a lot of superseding controls. What are those important versus the localized neighborhood importance of the forest I've been working in the small amount of soil and the forests and things like that? And I'll say, yeah, I would definitely agree that there are larger superseding effects that are responsible for this turnover, and where my research is intended to fit in are some of the unforeseen consequences that may come along with this change, right? Like the increases in pathogens that are also related to those other superseding global change factors, but this mycorrhizal shift is going to exacerbate them, right? And then also some of the reasons why things may not tip back. Yes, much of it is going to be due to these overarching global change consequences. Nitrogen deposition is a huge one. If you put more nitrogen in the soil from anthropogenic means, right, all the stuff we've been pumping into the atmosphere, that's going to favor arbuscular mycorrhizal trees over ecto trees because there's more mineralized nutrients that are avoiding that transformation through the soil saprotroph compartment to benefit AM trees. Whereas the ecto trees are still going to be relying on trying to get at that soil organic matter that's obviously not being deposited through the atmosphere for human means. Also selective logging, like you brought up, right? Things that really operate at the ecosystem scale that are causing these much bigger shifts. Those are definitely more important than the stuff that I'm exploring, for sure.
Okay, thank you so much. And let's give another round of applause.